Pues gracias a todos por vuestra asistencia al seminario extraordinario de hoy, que lo van a dar los profesores Filippo, Maggi y Giovanni Caprioli de la Universidad de Camerino, ¿vale? Va a ser un seminario compartido entre los dos, pero no va a durar más doble, ¿vale? Que no, no os agobiéis, que no es doble. Entonces ya va a empezar el profesor Filippo Maggi, que es titular en biología farmacéutica de la Universidad de Camerino en Italia. Forma parte del Centro de Investigación de Chemistry and Interdisciplinary Project. Actualmente es miembro del Senado Académico de la Comisión de Investigación de la Universidad y delegado del rector para el bienestar y el deporte. Es docente, abarca asignaturas como botánica farmacéutica, plantas alimentarias y fitonutrientes y cosmocéuticos. Su línea de investigación se centra en metabolitos bioactivos de plantas silvestres y cultivadas con aplicaciones en sectores nutracéuticos, cosmocéuticos y agroquímico. Es autor de más de 500 artículos en revistas internacionales, tiene un índice de 63 y actualmente coordina proyectos financiados por el gobierno italiano y la Unión Europea. También es editor asociado de Industrial Crops and Products y Plants y es uno de los científicos más citados del mundo según el ranking de Stanford y los High Ranked Scholars. Gracias.
Thank you, Susana. Thank you all for coming. It's a pleasure for me and Giovanni to be here in Madrid in this important research center and to show you the results of our research as a starting point for our interaction to start a new collaboration including the exchange of master students, PhD, joint projects, and so on. Okay, so I hope you enjoy my presentation. As Susana said, I'm a pharmaceutical botanist, but in the, let's say, the 10 years, I work on the developing of bioformulation, including natural products to combat different target insects, vectors, agricultural pests, mites, and so on. Okay, let's start. So, as you can see from the cover slide, this presentation is focused on two important medicinal plants. We dedicated, I think, the last 4 or 5 years of research, Carlina acaulis and Acmella oleracea. Our research were aimed at developing bioinsecticide formulation in the framework of the European strategy to reduce the use of synthesized chemical pesticides. For example, the farm to fork strategy of the European Commission aimed to reduce by 50% the use of these products by 2030. So why Carlina and acaulis? In the last 10 years, we screen a lot of plants, mainly belonging to 3 main botanical family: Asteraceae, Lamiaceae, and Apiaceae. Of course, there are also other plants like Cannabis sativa and others, but we focus mainly on 3 botanical family. Why? Because they are endowy secretory structure, so they produce essential oil. So the essential oil is one of the natural products we focus a lot, but not only. We work also on plant extracts and isolated compounds. And at the end of this period, we concluded these 2 Asteraceae plants are the most interesting for possible development of botanical insecticide. So I will give you an overview of the most important findings. And of course, we are a small part of a big network. Here I reported the main colleagues: Professor Giovanni Benelli, who Azucena know very well, from the University of Pisa, Dr. Roman Pavela from the Czech Agri-Food Research Center in Prague, and Professor Roberto Rizzo from the Research Center for Plant Protection and Certification in Palermo, Professor Alabolas Soulakis from the University of Palermo, and Professor Nicolas Cavalierato from the Agricultural University of Athens. So we carry out different studies on different target and non-target species. So let's start with the first species, Acmella oleracea, also known as jambu, is a Brazilian plant growing in the Amazon rainforest, but is also found in other tropical countries, and in the Mediterranean area in the last year is cultivated because it has several applications in pharmaceutics, cosmetics, dentistry, food and beverages, and we found out that it's very interesting also for the agrochemical industry. So it is endowed with several biological activities documented in literature, among which the most important one are analgesic, anesthetic, anti-inflammatory, insecticidal, and acaricida. So Acmella oleracea is reported as a botanical to be included in a food supplement in the Italian Minister of Health, and also in the Belfry list. Do you know the Belfry list included botanicals to be used safely in food supplements, for example. In those, it is also classified as safe by EFSA and by the Flavor and Extract Manufacturers Association. So this means that it's quite safe in order to possibly produce and develop a botanical insecticide. So this is the phytocomplex of the plant. You can see different classes of secondary metabolites, hydroxycoumarine, triterpenoids, phenolic compounds, the essential oil, but the most important class of phytochemicals are the normal alkylamides. These are biosynthetically derived from the condensation of unsaturated fatty acids with the decarboxylated amino acids. And the most abundant representative of this class of compounds is spilantone, also known as hafinin. It is endowed with 3 double bonds, and the most important bond is this, which is conjugated with the carbonyl group of the amide moiety. And spilantol has several applications in pharmaceutical, nutraceutical, cosmetics. Several patents are found. And what about the pharmacological, biological activity of this compound? It was found to be active on different vectors, such as mosquitoes, but also ticks and some agricultural pests. The main pharmacological properties of this compound are anesthetic, antinoseptic, anti-inflammatory activity. So they are the pharmacological activity of the plant, because spilantol is the main secondary metabolite, is the most abundant compound. And this pharmacological activity are linked to different pharmacological activities, for example, interaction with transient receptor potential vanilloid and ankyrin receptor, the blockage of the voltage-gated sodium channel, and the interaction of the GABAergic, serotoninergic, opioergic system, the downregulation of some inflammatory cytokines and enzymes, and the inhibition of transcription factor NF-κB. So we conducted some studies on the optimization of the extraction processes using conventional and more innovative facilities. You can see ultrasound extraction, SOx extraction as conventional, and supercritical carbon dioxide extraction, coupled with wiped film short pet molecular distillation, which is an innovative technique used in different industrial fields. So here you can see some results of concerning the spilantol recovery from the plant biomass using different solvent and different conventional extraction techniques. And from the other hand, you can see the spilantol concentration in the different extracts. You can see that methanol is the best solvent to recover the highest amount of spilantol from the plant biomass. Like, coupled with success, for example. This is the percentage from the plant biomass. But if you want to have an extract richer in spilantol, you have to use apolar solvent like hexane. That's why we focus on this kind of extract in the biological assays. And here you can see the results from one study focused on supercritical carbon dioxide extraction coupled with wiped film short pet molecular distillation. You can see that the supercritical extraction has a yield in terms of gram of extract out of 100 gram of extract of 5%. And the wiped film, the molecular distillation yield 60% of this 5%. But the composition of the extract derived from this technique we name alkylamides enriched fraction AEF is double in terms of spilantol concentration the supercritical through the extraction. So this kind of extract can be interesting from an industrial perspective because it avoids the purification of spilantol, which is time consuming, and this is very expensive in terms of solvent, consumables, and so on. This kind of process is without solvents. Of course, there is the machine, the equipment, but there are not organic solvent to be used. And in this study, we compare the mosquitocidal activity of the alkylamide rich fraction with respect to the supercritical extract and to spilantol, pure spilantol, which is isolated by column chromatography. And we observed that the alkylamide rich fraction has a comparable activity to that of spilantol. Why? Because it is composed of several alkylamides with similar structure to spilantol, which can give a synergistic effect to the extract. So you can see that alkylamides are the measure fraction of this extract, about to 62%. So concerning the pesticidal potential, this plant is less explored than the other one, than Carlina, but we perform some studies, other are ongoing. And in this study, in collaboration with Athens, we sprayed the hexane extract and the methanol extract on wheat grains, and the mortality was calculated on different solid product pests you have in your laboratory, have seen, and you can see the different mortality percentage displayed by different extract. In general, the hexane extract was more effective than methanol extract. Here you can see only data at after 7 days of treatment with the highest concentration of extract, and there were 3 insects: Alphitobius diaperinus, Tribolium castaneum, and Tribolium confusum, that were more resistant in the adult stage, but were neutralized at the larval stage by the extract. So the mortality was good. Here there are results from another studies measuring the sublethal effect of the hexane extract on Prostephanus truncatus, another pest. And at LC10 and LC30 concentration, they measure the duration and number of stops, of climb up, upside down, of insect. And as you can see from the table, the locomotor activities was significantly altered by administration of sublethal concentration. This is very important to study also the sublethal concentration of natural products. And this is the effect of LC30 on mating behavior and the mating the interaction of insect treated with sublethal concentration were significantly less than control. Okay, the safety profile of the extract and spilantol were measured in terms of cytotoxicity on human keratinocytes, the HACAT cell line. And the EC50 values were higher than 100 ppm for the hexane, the fraction of acrylamides, and higher than 50 ppm in the case of spilantol. So let's say that the extract is more safe than spilantol. And there are also literature data attesting the low risk of Acmella oleracea towards non-target organisms like Tetragonisca angustula and Solenopsis sevissima. Other studies are ongoing with the network I showed you before, and I think in 1 year we will have new results. So what about the mode of action on insect? Of course, there are no specific studies in literature, but from the pharmacological studies, we can hypothesize some possible mechanism of action. For example, the modulation of the GABAergic system can affect the central nervous system of insect, giving abnormal movements, spinning, uncoordinated muscular activity, like those observed in some studies. And also the property to inhibit the voltage gated sodium channel, which is a property like the pyrethroids, can affect the central nervous system. Other studies outline the property of Acmella extract to affect the histolysis and histogenesis inhibition, but specific studies are not yet reported. So let's move to the second plant, Carlina acaulis. This is, I think, more interesting for the effect we observe on different target insect. This is a typical plant of mountains, growing on the dry rock soil up to 2,000 meters of altitude. It is used in traditional medicine as a treatment of skin disorders, as an antibiotic, and to solve digestive problems. It is also used for food purposes as a wild artichoke. For example, in the Balkans, this plant is it is distributed in Central Southern Europe, mainly in Italy, France, Balkan, and East Europe. Also, this species is reported in the Belfry list, the root and the essential oil, in which we focus our attention, and also in the list of the Italian Ministry of Health for botanicals used in food supplements. This is the phytocomplex. The plants contain mainly in the roots caffeoylquinic acids, derivatives, triterpenes like ursolic acid, some flavonoids, and of course the most interesting part is the volatile part, which is the essential oil containing mainly a polyacetylene, which is carlinoxide. It is endowed with the aromatic and furan rings linked with a propionyl chain bearing a triple bond. And other volatile compounds occur in minor percentage in the essential oil, like benzaldehyde, R-curcumin, and beta-sesquiphenlandrine. So some studies were focused on the optimization of the extraction of essential oil and, of course, Carlin oxide, because we can consider the essential oil as a monocomponent essential oil, monocomponent product, because Carlin oxide occur at percentages higher than 94, 95%. We compare different hydrodistillation technique, conventional and microwave assisted hydrodistillation after a statistically based optimization. And we found out that microwave can enhance the yield of the essential oil. We studied also other parameters like the extraction times, the water biomass ratio, and so on. Here you can see the color of the compound of the Carlin oxide, and this is a section of the roots of the plant which are endowed with secretory ducts. This is the chromatogram, very simple. As I told you, it's a monocomponent, so very small peaks can be observed, but yes, we can say distillation is a purification method of the Carlin oxide. This is the mass spectrum. Okay, one of the first studies was carried out on mosquitoes and housefly, and we observed very low LC50 values. I think in literature this was one of the most active essential oil on mosquitoes. And also in this study we develop some formulation, microemulsion and nanoemulsion, and we calculated the LC50 of these products. And this was the starting point for other additional studies on target pest species of Carlin oxide and Carlin essential oil and formulation. This was conducted on Mediterranean fruit fly, Ceratitis capitata, which were treated with a mucilage, an emulsion made up of essential oil, Carlin oxide, mixed with dimethyl sulfoxide, and other ingredients such as a protein bait, sucrose, and carboxymethylcellulose sodium salt. This mucilage was placed in a bakelite cup inside the plastic container, and the ingestion toxicity was measured, and the LC50 values was around 1094 ppm. And afterwards, at the University of Pisa, measured the behavior of medfly treated with sublethal concentration of the formulation, and they observed a significant reduction of aggressive behavior of medfly. A similar study was conducted on olive fruit fly, Bactrochera ole. So they were fed with this mucilage, similar composition, and they determined the LC50 values for Carlina, acaulis essential oil, and Carlin oxide. What is the difference? Carlin oxide essential oil contains 95% of Carlin oxide. Carlin oxide is 99% pure. So there are minor compounds affecting the activity, but difference are not so significant. So the LC50 values was overlapping that on medfly in the case of Carlin oxide, 1052 ppm, and was a bit lower for essential oil, 700 ppm. So on this pest were conducted electroantennography studies to see the response of antennae respect to Carlin oxide and Carlin essential oil. Is you can see that Carlin essential oil elicit a bit higher effect on antennae than Carlin oxide. Some two-choice test by means of Y-tube olfactometer assay were conducted to measure the repellencies or attractiveness of the products, but this assay revealed that olive fruit fly is not attracted by these products, so no repellencies or attractiveness. So same or similar studies were conducted with Carlin oxide on food storage product pest. In this case, you can observe a higher toxicity also on some resistant adults stages like Tribolium castaneum, Tribolium confusum. Here you can see the two different doses, 500 ppm and 1000 ppm. And you can see that in most of cases, the highest dose was lethal, produce 100% mortality. The only exception was the adult stage of Alphitobius diaperinus. Okay, let's move to agricultural pests. Here you can see results from a study conducted on tomato pinworm, Tuta absoluta. And we use for the study a nanoemulsion made up of 0.25% of Carlin oxide, which was combined with a oil co-solvent, which is ethyl oleate. And then there is a surfactant polysorbate 8. So this is an emulsion was studied in terms of particle size, stability. And the test was conducted by spraying this nanoemulsion on tomato plants, which then were moved on 2 cups Harina experimental unit. And the ingestion toxicity assay put in evidence that after 24 hours, the nanoemulsion gave very high mortality in pinworm larvae. And after 12 days, the adult emergence from the surviving larvae were very, very low respect to negative control distilled water of the control nanoemulsion, which is the nanoemulsion without the Carlin oxide, the active ingredient. And also the inhibition on eggs hatching were measured, and this was very good, this inhibition, especially after 11 days post-treatment. In this study was measured the translaminar toxicity. In this case, the larvae were allowed to penetrate the leaves of tomato, and then the nanoemulsion was sprayed on the leaves. And after 24 and 72 hours, the nanoemulsion produced 100% mortality on larvae. This was very good results. Okay, these are the results on a vector of Xylella fastidiosa, which is Philenus spumarius, the so-called meadow spattered bug. And in this case, we develop an nanoemulsion containing 3% of Carlin oxide, combined with a wetting agent, polysorbate 8, and the oil co-solvent ethyl oleate. So the for ingestion toxicity assay, fava bean sprouts were used, where they were dipped in the formulation, and then they were put in the arena experimental unit in plastic cages, and they were exposed to the Philenus spumarius adults. and the toxicity was measured. You can see the group treated with Carlina oxide nanoemulsion were affected, so the toxicity was so high. Also in this case, some experiments by electron-technography olfactometer were carried out to measure the response in male and female antennae, and they detected that the females were more sensitive to the nanoemulsion, to the active ingredient, to Carlina oxide, than males. Here you can see the stability of particle size, the polydispersive index, and Z average over a period of 3 months. And in the anti-feeding assay, the nanoemulsion was spread on green beans, which were put in cups at the corner of nylon net cages, and in different time points, 1, 6, 12, and 24 hours, the presence of Philus spumarius was detected in treated and untreated green beans. And as you can see, treated beans um, were less occupied by the pest. So the presence of Philenus spumarius was reduced from 51 to 94%. So let's move to other target pest, aphids. This study was performed at the Czech Agri-Food Research Center. In this case, Carlina oxide was mixed with Twin 20 and sprayed on wheat plants where Metopolopium dirodum, which is one of the most important pests of cereals, was released, and different doses of the active ingredient from 0.5 to 3 milliliter per liter were sprayed, and they were compared with a positive control, which is a commercial product, Nudosan, which is a potassium salt of fatty acids, which is used against aphid. But the dose recommended is 20 milliliter per liter, so higher dose than those tested for Carlina oxide. And as you can see, that Carlina oxide at 3 milliliter per liter gave almost 100% mortality than the positive control at 20 milliliter per liter. And in these studies, also the subletal effect on fertility and natality were measured. LC30 and LC50 concentration were able to reduce the emergence of nymph from surviving adults. And the inhibition was about from 52 to 60%. Okay, let's move into another pest, the two-spotted spider mite, Tetranicus urticae, which affect many plants, horticultural and crops. And this experiment were performed on bean leaf disc placed in petri dishes, where the Carlina oxide diluted in acetone was sprayed using a Potter precision tower. The experiment were conducted on female, on nymph, and eggs. And as you can see, the Carlina oxide was more active on female than nymph, with LC50 values around 1000 ppm. And the mortality was monitored over a period of 4 days. And 5 different concentrations were assay from 300 ppm to 5,000 ppm. Of course, the highest concentration gave high 100% mortality after 2 days, when compared with the 2,500 ppm concentration that gave 100%, almost 100% after 4 days. And also the increasing rate of the population of the two-spotted spider mite was measured, and there was a very significant decreasing in this rate from the concentration of 1,250 on. And of course, the two highest doses were the more effective. Also in the studies, we observe a significant decreasing of oviposition rate as a function of the increasing concentration of the essential oil, and the fertility combined with the mortality data allow us to determine the total toxic effect, which was from 70 to 74% at doses of 625 ppm to 100% at the highest doses. Okay, what about the non-target toxicity, which is very important in the developing of botanical insecticide? Here you can see the mortality, the toxicity data on Daphnia magna, a aquatic microcrustacean. The LC19 concentration of Carlina oxide gave 44% of mortality, which is less than that given by alpha cypermethrin. Yes, I'm finished. And then we have mortality on predators or aphids, like Aphidoletes aphidimiza and Chrysoperla carnea. The mortality at LC90 concentration are quite low, and they are lower than that the nudosan that on Chrysoperla carnea produce 80% mortality. Other non-target pest, the phytochemistry mite Neoceulus californicus, which is a predator of Tetranicus urticae. No mortality was observed on adults. No side effect on eggs, but the only sign of the only side effect was a slowdown in egg hatching. And also another side effect was the repellency of Carlina oxide on adults according to the highest dose. So in the two choices test, we observe that at the highest dose, the phytochemistry escaped from the arena, and there were a higher number of mites on the disc with the lower concentration. So there is a repellence, but this repellence was short lasting. So after 24 hours expire. So this is quite good in the case of use of these products. Another side effect that can be taken into account is the phytotoxicity. Unfortunately, Carlina oxide can be toxic, for example, on tomato leaves. And in this study, we found out that using a formulation at 0.5% of active compound can give damage side effect on the leaves. Indeed, 0.25% of Carlina oxide can be safe. So phytotoxicity is a topic to be further explored because, as you know, natural products can be quite selective. They can be safe on some crops, can be dangerous on other plants. So mammalian safety, which is very important to market, to register a botanical insecticide. Carlin oxide was tested on rats for the determination of lethal dose 50, and the values about 1000 milligram per kilo of body weight is overlapping that of other natural substances like thymol, like cinnamaldehyde, like geraniol. So can be good to, and also some study on the tissues and organs evidences modest neurological and modest effects. This is a good point, let's say. Also on cell, the cytotoxicity was not quite high. Okay, what about the possible mode of action? We know that the propenyl chain of the compound can be the main moiety responsible for the activity of the compound, giving rise to carbocations, to radicals, causing oxidative stress in tissue, in organism. So then this oxidative stress can be enhanced by UV light, because polyacetylene are documented as a phototoxic compounds. And what about other action? Some hypotheses can be done, like the GABAergic interaction, and also the neutralization of the toxicant mechanism of insect, like cytochrome P450. But this can be further explored. So last slide. In conclusion, we have seen some strength and some weakness points of these two plants, Acmella oleracea and Carlina acaulis. What we can say about this? Of course, the pesticide potential is important, and also this plant attracted the interest of some agrochemical companies. The safety profile, of course, we didn't study many cell line, but it seems that they are quite safe. They are food plants. So what are the weakness points? In-field studies are lacking. Yes, we have done something on this plant with the agrochemical company, but the data are not available. And of course, more field studies should be done, but it's not easy. What about the supply chain, which is very important from the scalability of the product from an industrial perspective? Acmella oleracea exists under cultivation, so it's cultivated around the world, so no problems. Of course, the price of the raw material should be low. Carlina acaulis, this doesn't have agricultural chain. This means that the only solution can be the synthesis of the active compound, and we succeed in this, but the synthetic route is not evaluated as scalable from the company. What about the phytotoxicity? More studies are needed in order to determine the selectivity of this plant on different crops, and the same for the mode of action. We are doing something on Carlina acaulis, and let's say maybe we can have more data to support the mechanism of action. So that's all for this. We are available to study together other effects on other pests of these products. And if you have questions, do not hesitate to come. Yes, yeah. I also can send you the paper of this work. Of course, I've overviewed a lot of literature data. I'm sorry, but I have not so much time. So thank you for attention.